Hello, I'm Professor Matthew Schmidt, and I'd like to welcome you to Genetics. In today's session, we're going to discuss, under the general topic of chromosomes, we're going to talk about sex determination. And I arranged it this way for a reason. So we've now learned about the general properties and arrangements of chromosomes. We've understood how they move during mitosis and meiosis, and now we're ready to look at a couple of other interesting aspects of the idea that Mendel's genes are in fact located on chromosomes, right? Those ideas, I still want you to keep integrating them in your head. I know that you know this, but historically remember that the way we're talking about it, we've just put these two ideas together. So when talking about sex determination, while that is an important topic, we also want to highlight the difference of the X and Y chromosomes, at least in mammals, with all of the other chromosomes. And then in a subsequent lecture, we're going to see what happens when a gene happens to be located on the X chromosome. But let's start with sex determination. So it may seem like an obvious statement to say that most animals are either one sex or another. In fact, certainly that's normally true for human beings. We're either male or female. I don't know if it would surprise you to learn that in many cases this isn't actually true. There are many true hermaphroditic animals, and there's all sorts of strange things that can go on. But we want to start in the most simple way. So we're going to assume animals like a human being are one sex or the other. Cats are male or female. So the chromosome complement of males and females, let's just use humans, we both have 46 chromosomes, but the chromosome complement, and what I mean is a certain particular aspect of that chromosome complement, will normally determine whether someone develops as a male or as a female. So one thing we've alluded to before, but we've never directly said, is that in humans, remember we said out of the one way to look at the 46 is that there are 23 pairs, right? And it turns out that 22 of those pairs are what we call autosomes. Now we've referred to that word before, but autosomes, it doesn't mean they're not important chromosomes. They're vitally important for life. It just means that they have nothing to do with sex determination. And the autosomes don't differ in any appreciable way between men and women. It's the last pair, what by convention is thought of as the 23rd pair, that is the sex chromosomes. And there are two different types of sex chromosome. That's the only pair where that's possible. But even if they're different, we still consider that a homologous pair. So one pair of sex chromosomes, every human being has that. And the X and the Y are possible sex chromosomes that one might have. So we're going to figure out how this works. But probably you know that under normal circumstances, and just as a quick disclaimer, when I say normal, I'm not implying that people, as we're going to see some irregularities here, are abnormal. I just mean the way it usually happens, right? So normally, with that understanding, females, when it comes to the sex chromosome pair, have two X's. So actually, this is the least strange, if you want to think of it that way, because just like they have two number ones and two number twos, they have two X's. And those could have been numbered if we wanted to. In fact, you guys know that all the chromosomes pretty much look like X's. But they named it the X because of the possibility of the Y also being paired with it, and the X and the Y don't look very much like each other. The Y honestly really doesn't look that much like a Y, but that's what somebody thought it looked like, and it stuck. Males are XY. So males are interesting in the sense that that's a homologous pair right there that contains two different types of chromosomes, one X and one Y. Now the Y, it turns out, it's a very small chromosome. There are only a few genes on it. It's fairly well characterized, but we're going to focus on one or just a couple of genes there. The X is huge compared to the X. It's of normal size. What I mean by that, it's about the size of one of the larger autosomes, and it has many genes, thousands of genes, and those genes don't have anything to do with sex per se, okay? So think of the X chromosome as a relatively normal chromosome, and it's the Y that's sort of the strange one as things go. But whether you're XX or XY, obviously there are differences between males and females, but not in the sense of deep biological problem type differences. Okay? So a big question that we could ask, and I think it's an interesting one in terms of understanding the interplay between the sex chromosomes, is this. What actually determines the sex of an individual? Because there are two very equally valid hypotheses that you could come up with, right? So if we say, in rather simply, but we want to keep it simple here, if XY equals male and XX equals female, can you see that there's two ideas that you could come up with about what really causes an embryo to develop down the male pathway or down the female pathway? In fact, to look at them, embryos are completely gender neutral for about 2 weeks, and only after that do you see them sort of going down one road or the other. There are things going on in there, but I guess, so what I'm saying is this. You could make the idea, the hypothesis, that, the X's, we're not sure what's going on with them, but it is in fact simply the presence of that Y chromosome there that causes one to go down the male pathway. And that's logical because females don't have any Y chromosomes, so they become female. Males do have a Y. So one hypothesis is that it's the presence of the Y chromosome that causes maleness, if you want to say it that way. The presence of the Y. But another reasonable hypothesis is that the Y really doesn't do anything much, and that really it's the number of X's that one has that will make the determination about if they're going to become male or female. So that would be the dosage of the X. In other words, if you have 2 X's, you'd become female. If you have 1 X, you'd be male, and who the heck knows what's going on with the Y. These are both viable ideas. Now, there's a lot of evidence, and we do understand this rather well, but let me show you some evidence that will allow us to decide or see which of these hypotheses is supported. In humans, and I have to stress that while we're interested in humans, there are many different sex determination systems. Even birds, who are reasonably close relatives of ours, evolutionarily speaking, have a completely different sex determination model. So this pretty much goes for mammals, and humans in particular is what we're talking about. But just keep in mind that sometimes you'll see problems that bring up Drosophila or different types of birds that do have a different type of system than we do. So here's most of the early evidence, and it makes a lot of sense, comes from mutations or changes in which individuals actually have the wrong number of sex chromosomes. And what this means is you're supposed to have pairs, right? Whether it be XX or XY, you're supposed to have 2. But there are individuals who have 1, which is technically called a monosomy, and there are individuals that have 3, which is technically called a trisomy. So let's check out 2 of the most fundamental and reasonably common situations that can occur. There are individuals who exist who are XO. Now, XO means the O is just sort of like a placeholder. These individuals have 1 X and they have nothing else, so they really don't have a pair. Instead of 2, they have 1 at that location. Just to make sure you understand what I'm saying, algebraically, right? A diploid individual is 2n. If you're missing 1 member of 1 pair, you're 2n minus 1. In humans, that would mean that this person has a grand total of 45 chromosomes, right? So the situation where you only have 1 X chromosome and no other member of the pair is known as Turner's syndrome. And as I said, monosomy is a good word to know, meaning 1, but 1 in the sense of where there should be a pair, now there's only 1. So there's, in a sense, a dosage problem in that there's too little, right? You're supposed to have either 2 X's or an X and Y. All you have is the 1 X. But an important observation is that if you saw someone who has Turner syndrome, they definitely appear female. So keep that in mind. There is also a phenomenon where individuals exist who are XXY. They have three, and as I mentioned before, this is referred to as a trisomy. And a condition where individuals have an X, and a Y is known as Klinefelter syndrome, and those individuals most definitely appear male. Just to do this again, because it's a good habit and it's a good thing to understand, if you are trisomic for one pair of chromosomes, meaning you're supposed to have two but you have three for that particular chromosome, we end up with 2n plus 1, and individuals of that like that have 47 total chromosomes. Okay? Now, we see that we pretty much have the answer to our question here because of the following. Let me erase. Can you see, if I'm going to say it first, but then let's look at the evidence. In humans, it turns out it simply is the presence of the Y chromosome that says, if chromosomes can talk, Y basically says, look, go down the male pathway. That's the order I'm issuing. Being female is actually the default condition. So not having a Y chromosome, that basically means you're going to go and be female. Having the Y means you're going to be male. But how do we come to that conclusion? Well, if you look at the Klinefelter's individual, right, it really is, that person is the deciding evidence. One of our hypotheses were it depends how many X's you have, right? So that person has two X's, like a normal female does, and it also has a Y. So if that person appeared female, that would support our first hypothesis, right? That just the Y is irrelevant and the number of X's is the important thing. But in this case, the person appears male, and it seems that it must be due to the presence of the Y. Furthermore, if you look at the Turner syndrome individual, she only has one X, like a, quote, normal male. And again, that supports the idea that it's not the X chromosome that's making the decision, if you will, be male or be female. It's the Y. I'm not saying the X is not important, but it's not important in the actual signal that basically says you're going to be a male or a female. Now, if you look at the Y chromosome, we had mentioned before, it's sort of a tiny little thing. And the male gene, that's obviously not the technical term for it, but on the Y chromosome, there's a gene that's usually called SRY, and that refers to the sex-determining region of the Y chromosome. And right in that name, you can tell that that's the place on the Y chromosome where the decision is, you can almost think of it as flipping a switch and saying be male or not. No signal means be female. Some people call it the testes determining factor, but SRY is a little bit more common. Now, this gene, it's not 100% clear if it's one gene or more, but this region of the chromosome, it's pretty clear that it encodes one or more transcription factors. If you don't know what those are yet, that's quite okay. But it has to do with the expression of a lot of genes, most particularly enzymes that are active in making testosterone. So if you want to think of the simple version, this region on the Y, basically the signal that it sends is turn on testosterone. Testosterone is a steroid hormone that then has a lot of targets and turns on a lot of genes associated with maleness. So by saying being female is the default condition, what I mean is in the absence of any signal from the Y, things will proceed down the normal female pathway. Once the Y is present with its SRY signal, it basically says stop that. Now you're going to go down the male pathway. Now, that's, it's actually rather complex if you go through all of the molecular details, but I think that's relatively simple to understand. I had mentioned that birds have a different system, and we're not going to go into that in detail now, although I believe one of the problem-solving questions does talk about birds. Just to show you that it doesn't work the same all the time, Sex determination in Drosophila has been widely studied. It's well understood, and I understand Drosophila is not very much like a human being, but they're both animals, and they both have an X and a Y chromosome. They're not necessarily having the same functions as they do in us, though. So as you see here, sex determination in Drosophila, and there you have a picture of the beautiful fruit fly. It's beautiful to me, but you might have a differing opinion, especially if one's buzzing around your head. But it's not the same as in humans. But the reason I picked Drosophila and why it's so interesting is, as I said, even though it only has, it doesn't have a lot of chromosomes like we do, it has a grand total of 8, but there's one pair that's the sex chromosomes, and there is an X and a Y. In Drosophila, it's harder to, just look at one of them and, well, if you know what you're doing, you can tell pretty easily if one's a female or a male. But my point is, the Y chromosome does not have a similar function in Drosophila, and it is the opposite hypothesis that turns out to be true. We're not going to go through all the details of how that was determined, but it is the number of the X chromosomes that controls sex determination in Drosophila. More technically, it's the ratio of X's to autosomes, so assuming the right number of autosomes is there, it is the number of X's. So this is what I want you to take home from that. Don't worry about the Y in Drosophila. For this purpose, it's not doing anything, okay? So if you had a fly, we wouldn't call it Klinefelter syndrome, but certainly you could have a fly that was XXY. It would, in fact, be a female, assuming the autosomes were in the correct number. And that shows the difference, right? 2 X's, you get a female. 1 X, you get a male. So again, you don't use terms like Turner syndrome and Klinefelter's with Drosophila, but their equivalents would be reversed. And this is just what this really illustrates, is that sexual systems have evolved many times over evolutionary history, and they don't always work the same way. If you're interested in this, the molecular basis of sex determination in Drosophila is probably better understood than it is in humans, so you could look that up if you wanted to. Now, this also brings out a rather important point about gene dosage, because if you think about it, dosage really is very important under most conditions. Think about the autosomes for a moment. I mean, with autosomes, if you have a monosomy or a trisomy, there are usually severe problems. So the correct dosage for the autosomes, perhaps obviously, but you'll see why I say perhaps, is 2. If there's a deviation from this, we've got a problem. So the most common deviation is trisomy 21, Down syndrome, that we see in people. It's the mildest example of the effects of an extra chromosome. So people with Down syndrome have a total of 47 chromosomes based on what we said before. But the problem is that instead of the pair of 21s, they've got 3 21s, right? This is a dosage problem. There's too much expression of these genes on chromosome 21. And while many people with Down syndrome are high functioning and can live a good life, there are definitely problems in terms of, generally speaking, there are some mental deficits, generally speaking, but there are also physical problems. Often there are heart problems. The lifespan of someone with Down syndrome is less than the average for people who don't have it. Now, Down syndrome is interesting because it's really the only monosomy or trisomy of the autosomes that you'll ever see in an adult, because the rest of them are, quite frankly, so destructive to the developmental process that you don't even really see these. Sometimes children are born with these type of trisomies and monosomies, but it's really not a good thing, and they tend to, very sadly, to die early on. So my point is, if you go by that type of logic, you would think that the dosage is critical to be correct, to be 2, right? There's a reason we've got the pairs, and that's the correct number. If you go away from that, it's not good. But with the sex chromosomes, it seems to be a little different, because, we've seen someone who has Turner syndrome and Klinefelter's, a monosomy and a trisomy of the sex chromosomes. And while we didn't really discuss their characteristics that much, there are certain problems, notably sterility, uh, partial sterility. But in other words, these people can live pretty much fine. It's not biologically disastrous in the sense that most autosomal trisomies or monosomies are. So think of this. What about the dosage of the X chromosome? I mean, number one, you need an X chromosome to live, that's for sure. We said there are 1,000 genes on there that have nothing to do with sex. They have to do with, you know, general metabolic and physiological processes. You don't need a Y chromosome to live, obviously, because I have one, but any of you out there who are females, you don't have one and you're living quite fine, right? So we got to think about the X. Males have one, and if you looked at it from the male point of view, that might imply that, well, I have one, I have a few problems, but I'm pretty much fine. Maybe the correct dosage of the X is 1. It turns out that this is true, that the correct dosage of the X, it's the only chromosome that 1 is the correct dosage. Now, you could make an alternate hypothesis that 2 is the correct dosage, and then, you know, males are going to have a deficit. Either way you look at it, though, one of the sexes, based on everything we know about dosage, are going to have a problem, right? So I'm not going through all the experimental evidence, but it turns out that the proper dosage is 1. If 2 copies of an X chromosome were in fact expressed, it would be a problem. Females have 2 copies. Do females have severe problems because of this? I know a few uninformed guys who might say yes. I'm just kidding. But no, of course females don't have biological problems because of this, but we have to explain it. How can I have 1 X chromosome and women have 2 X chromosomes and all is good, when if you start doing that with the autosomes, it's a complete disaster? Well, it's a very interesting answer. Oh, that's the proof. Women do not have severe problems for having 2 X chromosomes. But why not? For real, guys, you would think that there should be some dosage inequity type of problem going on here. And it turns out that there's a phenomenon called dosage compensation that basically exists for the X chromosome and only for the X. Before we get into it, a lot of people, once they understand dosage compensation, say, well, we'll say someone had Down syndrome. They have 3 copies of the 21, right? Why can't we compensate for that? Well, it would be possible for that system to evolve, but it just didn't. The only chromosome cells know how to compensate for is the X. So what this means is, in every normal female cell, there are 2 X chromosomes. One of the 2 is shut down, and it's literally physically compacted. It's never heard from genetically, information-wise again, and it sort of condenses and hangs around in the cell. It's referred to as a Barr body after the person who characterized it. So in a very real sense, every human female is a mosaic for the sex chromosomes. Now, being mosaic, what that actually means, don't get nervous, and I'm not kidding. Some people find this a little odd. If you're a woman, it means that roughly half of your cells are expressing the genes on one of your X chromosomes, and roughly half are expressing the genes that are on your other X chromosome, because it sort of randomly gets turned off early on in development. So it's very interesting, but clearly this doesn't really have any noticeable effects in a human female. In Drosophila, just because you know the difference between the human and mammalian sex determination system and the Drosophila system, it turns out the proper dosage is 2. And I only mention that to show you once again, things don't work the same way in all organisms, at least in this respect. So an opposite thing is done. The correct dosage is 2, and to make up for that, now instead of females shutting one down, males have to hyperactivate their X to ramp up the expression of that genes on the X chromosome so it gets to the proper dosage. Now, just so you understand this, if a human individual was triple X, and that does happen, 2 Barr bodies would be formed. Guys, the cells have a very definite way of figuring out how many X chromosomes are in the cell, and any in excess of one get shut down, get turned into Barr bodies. So it wouldn't happen, but I'm just saying. Say you had 8 X chromosomes in a cell. In theory, 7 of them would be shut down and turned into Barr bodies. 1 would remain. That would be the correct dosage. Now, interestingly, cats are often used to try and understand or experiment with these sex chromosomes because you can see the mosaicism more clearly in female cats of certain types, and the reason is because their coat color gene, or at least one of them, resides on the X chromosome. So there's a calico cat you may have seen. Calico cats have patches of different colors, usually like almost an orangish color and a white and black. So that's mosaicism. Calico cats are always female, you guys, because it results from the random inactivation of one X or the other. And if you really get into it, it gets a little bit more complicated than that. But that means that there's no such thing as a male calico cat. One exception to that, and you can look this up. It's like a riddle. How could a male cat be a calico cat? The only way is if that cat was the equivalent of a Klinefelter's human. In other words, if a cat was XXY, a Klinefelter cat, if you will. I don't think they use that terminology. But one of the X's still gets inactivated, right? So even though the cat's going to look male in its sexual appearance and biology, it still has this X inactivation going on. So we learn a lot from the Klinefelter type situation. One other thing I wanted to mention before we conclude this discussion is someone asked me once a long time ago and I couldn't answer, and this is not, believe it or not, 100% understood. But think of someone who has Turner's syndrome, right? They have one X chromosome. We just got done saying that 1 is the correct dosage for the X. So then what the person asked me was, so then why do they have a problem? They have one X, that's good. They don't need a Y, they should be perfectly fine. And it really is a good question. So the best answer I can give you is that the process of X inactivation does not occur right in the zygote. It occurs early on during development in the first few weeks. But something about that early development and the imbalance of the X chromosome is where whatever problems exist come from. One other thing I should mention is that it's not just having one X or more, having a monosomy or a trisomy. Another problem is, if you think about what we learned about meiosis last time, when you have an odd number of chromosomes, remember 45 or 47 in humans, there is a problem with meiosis in that gametes become imbalanced. We want in meiosis one member of each homologous pair to go into each gamete that's being formed, right? That's what segregation meant. If there are 3, gametes inevitably end up with 2, and that's an imbalance. If they join with another normal gamete, you're going to get another individual that has a problem. So a lot of times the problem just have to do with the fact that during meiosis, if it's a sterility problem at least, that's part of the issue. But there's something going on very early in development where this has to unfold in the proper way, and that explains whatever other problems occur with individuals with, say, Turner's or Klinefelter's syndrome. But it's an interesting question, and it turns out that individuals with XXX syndrome don't have a terrible lot of problems. So if you were thinking about that, that's a good question. Okay, well, that ends our discussion on dosage compensation, but I want to give you a little preview. One of the reasons I put it here is because the next thing we're going to talk about is what if a regular old gene that has nothing to do with sex ends up being on the X chromosome. Remember, it's a regular old chromosome that you need to exist. It shouldn't matter, and in many cases it doesn't. But because of the fact that males have one X and females have two X's, there are some special rules or special patterns of inheritance for genes that end up on the X chromosome. And that's what we're going to explore next time when I hope to see you back at another genetics session.